Good morning, everybody, and welcome to this webinar of MedAID Work Package 3. The title of our Work Package 3 is Genetics and Breeding, and today we want to present the results of work we have done in this project for the last few years. My name is Sonesson. I work for NOFIMA. And as you can see on the screen, in this work package, we have worked with especially two industry partners. That's Colmarex and from Spain and Galaxidi from Greece. And as research partners in this work package, we have had Wageningen University Research, Roslin Institute and INIA and University of Ege. First, I would like to give you a couple of slides about selective breeding, because that's the, let's say, the core of this work package. So the FAO, they had a big state-of-the-world aquatic genetic resources for food and aquaculture report that came out in 2019. And that was a big step for aquaculture genetics. And it's in that report, we can read that there are more than 600 aquaculture species in the world. However, only 40% of these were produced from, and 40% of these were produced from unimproved wild types. It means that 60% are then from improved wild type, improved stocks. And 45% of the countries noted that genetic improvement is still not significantly impacting aquaculture production in the countries. And that's actually a bit surprising, I think, because we know that this is a very powerful method to have increased production. So Jedre et al., they found out that when they looked at all species that were available in 2012, the genetic gains that these different types of breeding programs, still they report about 12% genetic gain per generation. And we know from our studies later that if we can use genomic information in genomic selection, then we will increase gains with 25 to 50%. So it means that this selective breeding is something that we want to have better and more of in the world. We also know that genetic gains is actually giving money to the industries that are introducing it. So it's been valued at 0.2 euro per kilogram fish after one round of selection, both in sea bream and in Atlantic salmon. So what is the process of selective breeding? So if we look at the picture to the left, it's the simple schematic view is that we are mating broodstock, and then we select the best offspring of these to become parents of the next generation. And what we mean with best, that's what we have in our breeding goal and in our selection index. So that is typically growth, it can be disease resistance, and other traits that are important for the industry or for the population. What's also important to know is that selective breeding is cumulative over generations. It means that if you set up a breeding program, you have to be able to run it for several generations to have real gains in it. Although the gains comes immediately, but it will be more over generations. And here is what the value of genomic selection and why that is coming. And in the genomic selection increases the accuracy of selection. And what we are doing in the breeding program is that we genotype the parents with a race that we are going to hear later about. We also, and they are at the top, these parents, they produce two types of offspring groups. So to the left, we have one offspring group that we call the test offspring. They are phenotyped and genotyped. And we need at least 10 to 20 series per family to estimate the effects of each genetic marker of each SNP. Then we all, to the right, we have the second group of offspring. They are the candidate group. They are also genotyped, but they are not necessarily phenotyped. They can be phenotyped as well, but they don't have to be that. So they are only genotyped. And then we use the marker effects that we estimated in the test offspring group to calculate breeding values and select parents from these. And since in the aquaculture world, we often have large families, we can actually have a high within family selection in addition to the normal between family selection that we are doing. So that's where the gain is coming from. Then just 2 words about the selective breeding in the species that we are studying in MedAID. That's the European sea bass. And in 2016, it was estimated that approximately 50% of farm sea bass came from selective breeding programs. I don't really know what the figure is today, but it's probably increased a little bit. To the right, we have the gilt-tip sea bream, and the first commercial breeding programs for sea bream were initiated in the early 2000. Yeah. Good. Now we're back to the WP3. And here are the objectives of this work package. The first one is to develop a combined SNP array for sea bream and sea bass. The second objective was to develop and test genomic selection to improve key production traits in sea bream and sea bass breeding. The third was to discover individual genes associated with production traits in the species. The fourth objective was to characterize the genetic diversity of representative populations of the species and to test measures of inbreeding. And the fifth and final objective was to assess the existence and extent of genotype environment interaction for temperature on production efficiency for sea bream. So now I will present some slides on which knowledge and technology development gaps has MedAID WP3 filled, and a short version of the conclusion and recommendations. After this presentation, you will hear each of the task leaders. They are presenting their task with more extensive conclusion and recommendations as well. So first, a couple of words about the SNP array work. So the high throughput genotyping arrays, the SNP arrays, are essential tools for genetic studies. They are used for many things. We use them a lot for parameter estimation in selective breeding and for gene mapping, genomic selection, and to estimate co-ancestry between and within populations and other population genetics measures. And when MedAID started, and since then they have increased, but there were already existing some SNP arrays for important aquaculture species: salmon, rainbow trout, common carp, catfish. Now we have white-legged shrimp and tilapia. But when MedAID started, there was no SNP array available for European sea bass and sea bream. However, from other projects, mainly funded by EU, but also others, of course, there was already an extensive database of SNP markers available. But so in this project, we have developed a combined SNP array for sea bass and sea bream. It's called a MedFish array, and it was developed together with Performfish Consortia, which is Performfish is a sister project to MedAID. So conclusion and recommendation for this part of the work is that indeed we have this SNP array available now. It was created in collaboration with the Performfish Consortia. It has 60,000 high quality SNPs. They are split between the European sea bass and gilt-tip sea bream. But they were derived from evaluation of over 20 fish populations of each species. It means that we hope then that it can be useful in many populations, valid there. And it has already been used, and you will hear later about some results where it how it performs. So it's been used in our project and in the Perform Fish project, but also in populations external to these projects. And the second part of the work that we are going to present is on a genetic study of lipid traits. And lipids are linked to production efficiency, health, and product quality. And excess dietary lipids that are not deposited in the edible muscle or used as energy source for growth, they are considered a loss and an indicator of low efficiency. And also, lipids are important for the health of the fish and human. And for the fish, it is if they have excessive lipid deposition in and around internal organs, and there's an increased risk of metabolic disorders, oxidative stress, and inflammation. And of course, lipids are important for the consumers because fish lipids have omega-3 fatty acids that you have probably heard about, that they are good for you. You will hear more about this as well. So here for this work, we estimate the genetic parameters, did some gene mapping, small-scale gene mapping, calculated the genomic selection accuracies for lipid traits. And the important traits there are total fatty acid contents, total fat and fatty acid contents in the whole body, muscle, and liver. And some short conclusions for that work is that we saw a high potential for selective breeding on total fat and fatty acid content traits. For example, we saw medium to high heritability estimates, and we saw an corresponding high, medium to high accuracy of selection. We used the distal meter to measure fat, and we saw that that worked well for genetic studies, but less good for nutritional studies. And we saw some candidate traits in one of the species, and not so significant in the other species. And so we conclude that, let's say, the gene mapping work, for that we needed a larger sample size. I think we also need better genomic resources to actually find a better annotation of the genes, for example, so that we can get better studies on that for these two species. Next part of work is on population genetics. So why do we need this kind of work? Is one important thing for breeding program is that we want to use genetic information to set up base populations in a good way. And that is because genetic variability in the base population determines subsequent genetic progress. So if we start with a very narrow base, there's nothing to develop on, let's say like that. This base population can be created from both wild and farm populations. And to have an informed setup of your base population is necessary to determine the relatedness both within and between the populations. Population is also population genetics is also important for to manage genetic variation in an ongoing breeding program. What we actually talk about there is to control inbreeding. It's also good to know the effective population size in currently and what was in the past. And that's needed to ensure the sustainability in the long term. It also has to do with this narrow and broadness in the population. The third one is that it's good to have information about both the farmed and the wild population on their genetic relationship for wild and farm and wild fish interaction. Fish escapes are perceived as a major threat to natural biodiversity. We don't want to have escaped fish into the wild, of course, and knowledge on the relatedness between wild and the farm populations can be used in risk assessment for this, to see how separated the populations actually are. So what we did in MedAID here was to estimate genetic relatedness within and between farmed and wild populations in the Mediterranean. And short conclusions on that work was that the wild populations within a species, they show low levels of differentiation, whereas the farm populations, they mark larger differences between them, and they also differ from the wild populations. So you can actually separate these two groups of populations. It means that it's even more important then to avoid escapees, because crossing with the wild population could have undesirable genetic effects in the native populations. This information can also be used for new breeding programs. So we actually have informed selection for them. In the farm populations, there were often low estimates of effective population size, and that means that these populations need to apply selection and mating approach designed to control inbreeding and therefore loss of genetic variability to ensure sustainability in these. Finally, we have genetic by environment interaction. This was studied in sea bream. So what is genotype by environment interaction? It's the phenomenon that different genotypes perform differently in different environments. It's often measured as the correlation of breeding values estimated in 2 or more environments, and we can see re-ranking of families, how they perform in 2 or more environments. And when there is G by E interaction, it means for the breeding program that selection strategies need to be adapted, or you need to actually separate the breeding populations into different environments in order to get the genetic progress in each of these 2 environments. And in the case here, it was studied temperature was the environmental factor, and we know that with climate change, the water temperature might increase, so this G by E problem might actually increase in the future. And in WP3, there was a G by E study of sea bream for temperature on production efficiency. So what came out of that work? So they saw strong G by E for harvest weight, fillet weight, and growth rate of sea bream between East and West Mediterranean sites. But it was limited for fillet fat percentage. And because they found this G by E, now this company that were having the population need to think of a strategy how to select and test fish in both environments, and maybe to get information into their breeding value estimation. Maybe they can also select which family should go to which environment. And then again, we believe that this the importance of G by E will increase in the future if the climate change is actually changing the environment that our fish are held in. Thank you. That was my part of this.